calling Mr. Pierre Valiorgue from Eberna in Rhine in Toulouse, going to talk about CO2 bioconversion efficiency.
Thank you. Hello, my name is Pierre Valiorgue. I'm starting the last year of my PhD in LMFA Laboratory of Fluid Mechanics and Acoustics in Lyon, in INSA Lyon and Université Lyon. And I'm going to present a part of my PhD which has been done in collaboration with Microphyt, that some of you may have visited on Monday, and with Erwan Vermel, Dany Pradel, Laurent Beau, and Arnaud Muller-Fegat. So the part that I will present is concerned with CO2 biofixation efficiency of Microphyt photobioreactors. So I will briefly reintroduce Microphyt, this project, and then a dissolution model and how to complete the dissolution model in order to predict the CO2 contained into the biomass. So currently, 2 photobioreactors are operated in Microphyt in Valiorgue. They are placed into east-west oriented greenhouse, and they have been especially designed in order to grow slow-growing and fragile microalgae. So in order to reduce the mechanical stress within the photobioreactors, they are operated with a low-pressure pump, and the usual gas column used in order to realize mass transfer has been removed, and the mass transfer is now realized within the photobioreactor by concurrent two-phase flow of gas and liquid. So the CO2 is injected punctually, and it dissolves along the photobioreactor and brings the carbon where it is consumed. And in order to remove the excess of oxygen that is inhibiting the microalgae growth, air is injected continuously along the photobioreactor. Okay. So first, in order to study the dissolution efficiency, which is defined as a dissolved mass over the injected mass, we've been looking at the dissolution of an elongated bubble immobilized within a 6 meters long duct under a constant water aid. And this work has been published in Chemical Engineering and Processing. So we obtained a formula here for the dissolution efficiency of the bubble, mainly based on 2 hypotheses, considering that we have a flat exchange interface and a constant bubble height. And those 2 hypotheses have been checked and validated with this graph here, plotting the log of the bubble length over the initial length as a function of the contact time. So, yeah, the contact time is a very important parameter for the dissolution. So as you can see here, we obtain different slopes for different Reynolds number. And those slopes are related to the mass transfer coefficient that we can estimate and plot here in dimensionless form. So the mass transfer coefficient is using the Sherwood number, and we plot it as a function of the Reynolds number, which takes into account for turbulence, and as a function of the Schmidt number, which takes into account for the nature of the gas. And we've shown that our data are very well correlated by correlation found in the literature given by Lamourel, which was measured for wetted wall columns. So they also had a flat interface with small waves. Okay, so that's the basis of the work. Now we want to adapt this formula to the flow in the photobioreactor. So the third hypothesis is to consider an equivalent stratified two-phase flow, and so that we can express the contact time as a function of the gas section. Here is a section of the stratified flow in a pipe, and as a function of the gas flow rate. The interface can be expressed as a function of the liquid I, so that this expression here is a function of liquid flow rate, gas flow rate, and liquid I. Classical analysis for two-phase stratified flow relates the gas flow rate, liquid flow rate, and liquid high, so that this formula here is now only a function of process parameters, gas flow rate, and liquid flow rate. Okay, so now we wanted to test this formula. So on this plot, we've plotted the dissolution efficiency as a function of the photobioreactor's length. Each dot, the plus dot, represents the model for 10% concentration in the injected gas. And each dot is one length, one rectilinear part of the photobioreactor. And it compares well with the measurement here of 72% for dissolution, measured with concentration measurement of the injected gas and of the output gas. So we can see that the dissolution is not a strong function of the concentration gradient for 10% concentration in the injected gas, but it's much more, the concentration gradient varies much more for 20% here. So the dissolution is less uniform for 20% in the injected gas, but the final dissolution efficiency at the end of the photobioreactor is better for 20%, of course. Okay. And the other study we can, the other results we can have from this formula is looking at the effects of how we can tune the flow rates in order to optimize the dissolution efficiency. So those parameters here are the only one depending on the gas and liquid flow rates. So it has been plotted here, and we can see that it is maximized here when the gas flow rate is minimum, and this is mainly due to the fact that it increases the contact time, and when the liquid flow rate is maximum, mainly due to the fact that it increases the turbulence. Okay, so now we have a model for the dissolution efficiency, and we can predict how much of the CO2 is dissolved into the liquid. But if you want to predict the biofixation efficiency, which is the one really relevant, how much CO2 is contained into the harvested biomass over how much CO2 has been injected, we need to look at the stripping phase, which is used in order to remove the excess of oxygen, but unfortunately also remove CO2. So if you apply a mass balance on this phase and divide it by the injected mass, we have a relationship here for the efficiencies within the photobioreactor. So we have neglected the residual efficiency that takes into account for the mass in the liquid at the end of the test. It's approximately 10 grams, and the mass biofixed is 36 kilograms, so it's justified. So we've talked about the dissolution efficiency, and we've modeled the stripping efficiency due to the air injected removing the CO2, considering that we have an equivalent, as it is continuous, we have an equivalent rectangular exchange interface and an equivalent interface with along the photobioreactor. And we can see that it's proportional to the time of production over the mass injected. And this is a huge simplification because all of the parameters that are light, concentration of gas, of microalgae are taken into account within this parameter. So that measurements on the photobioreactors are much more simple. So those two parameters, those two dissolution efficiency models are function of the tube length. So we thought we could address the scaling of the photobioreactor and simply with a differentiation with respect to the length, we can express an optimum length of the photobioreactor as a function of the dissolution efficiency, biofixation efficiency, and tube length for corresponding to the measurement of those two. So we need to measure the biofixation efficiency with the dissolution we've done it before. And we've done it for neochloris oleoabundance, for which we know the chemical equation and the simolar mass as it has been measured by Mr. Privo. So from this simolar mass, it is possible to estimate how much CO2 is contained within the microalgae. So by weighting the produced dry weight of the microalgae, we have how much CO2 is contained, and dividing it by the injected mass that is measured by weighting the CO2 bottles between the start of the process and the end of the process. So it's measured over 3 weeks, 22 days. And we've also been careful to have approximately the same cell density between the beginning and the end of the process in order to diminish the residual parts. And the biofixation efficiency is evaluated to about 36%. So if we plot it if we plot the interpolated model through those values of dissolution here, which is plotted on this axis, and transformation or biofixation, sorry, I should have changed that, on this axis, the stripping efficiency is obtained by the mass balance, and is plotted on this axis, but we should be careful, it's in the other way. We can see that we have an optimum length for the photobioreactor, maximizing the biofixation efficiency, which is between 1 kilometer and 1.4 kilometer. And the other, so here there is an hypothesis that the concentration gradient does not vary within the photobioreactor that has been checked before. And we can see that there is two asymptotes, one here with dissolution and one here with stripping. So I guess I've said everything about this graph. So as a conclusion, we have a model of dissolution as a function of the process parameters. And we've seen that the liquid freight is mainly related to turbulence for dissolution, that gas freight is mainly related to contact time, and we've seen the effect of CO2 concentration. When it rises, it's less uniform. If we want to be able to predict biofixation efficiency, we need to take into account for the stripping phase, and it enables optimization for this objective. We've not finished to harvest all the results of this equation. And this equation here from my lectures from yesterday, I thought we needed to take into account for the momentum that could damage the cells, for example, momentum from turbulence, but apparently it's more the gas interface that damages the cell and less turbulence. Thank you for your attention, and we may answer your questions.
Question? Yeah.
You have said you remove the excess of oxygen. What value do you consider as an excess in % of oxygen in the gas?
Yeah, we have to work on that. There will be two objectives, optimizing biofixation and optimizing production, because we have to produce. This excess of oxygen is the one that is a constraint for production.
Well, it is a constraint for biomass production in terms of quantity, but in terms of quality, it's not necessarily a constraint, because if you have an excess of oxygen, you have a lot of antioxidant product, which gives a higher value to the biomass you produce.
I would like to understand, if possible, exactly the utility of your formula relating the optimal length of the tube with the length of the tube and the CO2 consumption efficiency. Because you, in your formula, the length of the tube is related with the length, the optimal length of the tube is related with the length of the tube and the efficiency. The efficiency is a function of the flow rate, liquid flow rate through the tube, the gas flow rate through the tube, and the CO2 concentration in the gas. It's true? It's true.
It is a function of that.
So, my feeling is that the utility of your formula, because the length of the tube is fixed. The length of the tube is the microphyt, the tubular reactor have a determined length. Okay? This is fixed. So, my feeling is that your formulas is useful for managing the liquid flow rate, the gas flow rate, and the concentration, because the optimal length should fix the length of the tube. The optimal length of the tube should be equal to the length of the tube. So this is my is true my thought or not?
Yeah. Actually, at the moment, the photobioreactors length is fixed, but we could imagine, and for next photobioreactors, well, first of all, we had no idea before that what was the optimum length for photobioreactors.
Of course.
So that's one information.
Yeah.
And it could have been useful to change that and adapt that for next photobioreactors. But we also can think that this is also a function of the growing rate of the microalgae. So if we change the growing rate for a different microalgae, we will obtain a different tube length. So in our next photobioreactors, we should think about being able to change this length of photobioreactors.
Yeah. So your formula is your equation is a Dessin equation. But now, because the Dessin is fixed, you are using your formula for managing the liquid flow rate, the gas flow rate, and the CO2 concentration. Thank you very much.
Agree.
Question?
Olivier Bernard from INRIA. Key parameter when dealing with carbon inorganic dissolution is alkalinity. And I missed something, but I didn't see where it came to the game in your model, especially for algae that are that can modify alkalinity.
Yeah, all of those parameters are taken into account in the production time. So we have to have an idea of the production time. And the production time is for the steady state. So when the concentration, this is easy in Microphyt's photobioreactors because there is a feedback loop, for example, on CO2 for a set to set the pH. So it works well. We have a very good steady state on Microphyt's photobioreactors. So I guess if you're producing this type of microalgae, you have to wait the steady state, measure the production time, and then you can inject it in the model. Have I answered your question? No.
Last question.
Pascal Guiraud, INSA Toulouse. I think it's quite the same question. I would want to know if you account for the chemical reaction of CO2 with water in your model, because the CO2 transfer is increased by the chemical reaction of CO2, and I don't know if it is accounted for in your model.
The CO2 transfer is increased by, sorry?
The chemical reaction with water. It is well known.
The CO2, well, what is important for the CO2 transfer is the CO2 gradient, and it's much more important. It's like 400 times greater than the concentration in the gas. When we are using 10% concentration in the gas, the equivalent concentration in the water is 400 times the one in the gas is 400 times the one in the water. So yes, it's a function of that, but do you agree?
I agree that the concentration in the gas is much more important than the concentration in the liquid. It's okay, but
so if it changes in the liquid, it will be very small compared to the if it's about 10%. Yeah, this model is not valid for very low concentration in the gas.
But you have equilibrium between the gas and the liquid at the interface. That's okay. I think it is accounted for in your model, but you have chemical reaction inside the liquid that consumes the CO2. Okay, so we will discuss later.
I think you will discuss at the coffee break, and I think it will be interesting or during lunch. So thank you very much. I'm calling Mr. Fabrice Gascon Villagoma.